Hello everyone. I'm very pleased to participate to this online event and to have this opportunity to talk about our recently published study entitled "Where is the Optimum? Predicting the Variation of Selection Along Climatic Gradients and the Adaptive Value of Plasticity." So this is a case study on tree phenology. Why asking such question for tree phenology? Because the advancement of spring phenological event in plants is probably one of the most documented examples of the impact of recent climate change on ecosystem functioning. And studies reporting this temporal trend have found that most of this response is due to the plastic response of plant phenology to increasing spring temperatures. But we often ignore if the plastic variation of phenology allows tracking the variation in optimal trait values. So whether plasticity is adaptive or not. And if plasticity does not allow to perfectly track the variation in the optimums as represented here, then we can expect genetic evolution to play a central role in the response to climate change. Many theoretical models predict when evolution and phenotypic plasticity allow adaptation to changing environmental conditions. And this model often assumes stabilizing selection around an optimal trait value, and that environmental change only affects this optimum. But in many species, and especially in long-lived organisms, we often lack the empirical data to parameterize models of phenotypic adaptation. And for this reason, in complement to empirical estimates of these important evolutionary parameters, we propose in our paper that mechanistic models can also be used to investigate the adaptive value of plasticity and the ecological causes of selection. So what we propose is a three-step approach that can be used with any mechanistic model that describes the causal relationships between environmental conditions, phenotypic traits, and fitness. And in our specific case study, we use the phenotypic or mechanistic model PhenoFit that has been developed for temperate tree species. And as you can see here, this model takes as an input a local climate and a parameter set that describes the reaction norms of developmental rates for vegetative and reproductive organs to climatic variation. And the main assumption of this model is that the timing of developmental events and the seasonal variation of climate affect individual fitness. So we first perform a single calibration of these reaction norms using large-scale observations, so that the variation in the output, so the variation in the predicted phenological dates and in the predicted fitness, is only due to the plasticity of the trait that is captured by this reaction norm and by the PhenoFit model. I don't have much time to go into details, but then we also use extensive empirical data to validate the PhenoFit model. And finally, and most importantly, we use this model to simulate fitness landscapes. So first, we use the adjusted reaction norms of bud growth rates to temperature. That is the main physiological response driving the variation in bud burst date. And we use this reaction norm and a given climate to predict a local bud burst date and the local fitness. But instead of using just one simulation with one parameter of the reaction norm, so here the mid-response parameter, we vary this parameter to generate variation in the bud burst date predicted and in the resulting fitness. And so by performing this sensitivity analysis of the model, we managed to retrieve the relationship between the bud birth date and the fitness, that is the fitness landscape. And from both the fitness landscape and the local date predicted with the adjusted reaction norm for a given climate, we can then estimate the resulting selection gradient. So we developed this approach for 3 different European tree species. Here, European beech, Fagus sylvatica, Cecil hawk, Quercus petrae, and silver fir, Aedes alba, species which are co-occurring in the Pyrenees mountains and which are intensively monitored along 2 elevation gradients in the Gave and the Osso Valley. So we use this empirical data to calibrate and validate the phenophyte model for each species. And to run our simulation, we use reconstructed historic climate for these 2 elevation gradients from 1960 to 2012. So our aim here was to derive fitness landscape in temperate tree species using a mechanistic modeling approach to understand how the optimum and the shape of the fitness landscape change with climatic variation, whether or not the plastic response of bud burst phenology allows tracking the variation in the optimum, and what was the resulting variation in selection along elevation gradients. The main output from our simulation work is a simulated fitness landscape for each species and each elevation along the elevation gradient. So from low elevation in red to high elevation in blue. And we can clearly see here that we predicted a strong variation in the shape of the fitness landscape between species, but also between elevations, with a change in the optimal trait values of this fitness landscape, but also in the width of the fitness peak or in the maximal fitness predicted. So focusing more on the variation of the predicted plastic dates, here represented with the red crosses, and the variation in the optimal bud burst date, here represented with the black dots, we can see that we predicted that both the plastic and optimal bud burst dates increase with increasing elevation in all species. And we also found that plasticity is not perfectly adaptive, so it never really managed to perfectly track the variation in the optimum. But we can also see that for beech and fir, these predicted dates are often included in the range of trait values that provide 95% of the optimal fitness. So they are very close to the optimum. So for that reason, we can say that plasticity is more adaptive for beech and fir than it is for oak. If we represent the variation in the phenotypic mismatch with elevation for the 3 species, we can see similarly that the average phenotypic mismatch and its variation in fir here in green and in beech here in black are quite small. And this phenotypic mismatch is much higher for oak here represented in brown. And also that the phenotypic mismatch increases a lot with increasing elevation in oak. For all species, we predicted that the width of the fitness peak decreased with increasing elevation. So we predicted narrower fitness landscape at high elevation. And this pattern is quite consistent among species. And finally, from both this variation in phenotypic mismatch and in the width of the fitness peak, we predicted stronger selection toward earlier bud burst date with increasing elevation. And this is particularly true for the 2 deciduous species, so beech here and oak. So to summarize the main result of these studies, we found that the plastic response of spring phenological traits helps 3 species adapt to climatic conditions, but that most often this plasticity was not sufficient to perfectly track the variation in optimal trait values, which resulted in selection for increased precocity with high elevations. We also predicted a strong variation in the shape of the fitness landscape between species, but also between elevations, so between climatic conditions, with fitness landscapes that were more often asymmetric and with width of the fitness peak and maximum fitness that varies a lot among elevations, especially for population close to their upper range limits. So all these results suggest that focusing only on the change in optimal trait values and neglecting other changes in the shape of the fitness landscape may be misleading about the role of plasticity and evolution in heterogeneous environments. And with that, I would like to acknowledge the contribution of Ophélie Ronce, Isabelle Chuine, Bertrand Teuf, and the other co-authors to this work. And I would like to thank you for watching this talk. And feel free to contact me if you have any questions or to have a look at the paper for more details.